Okay, now let's start with the histopathology fixation session. So question number one. Which substance is commonly used to fix phospholipids in tissue sections? And the answer is osmium tetroxide. Next question. What is the optimum pH range for fixation? 6 to 8. Which fixative is a gas dissolved in water and polymerizes in the cold? Formaldehyde is the answer. Which is the most commonly used fixative in routine histopathology? 100% neutral buffered formalin. Which physical method of fixation uses electromagnetic waves? Of course, it should be microwave fixation. Which fixative is best for preserving mitochondria? Potassium dichromate. Which fixative is commonly used in cytopathology as in case of spray fixation? It's 95% alcohol. What is the main disadvantages of using mercury chloride as a fixative? It leaves black precipitates in tissue. That's why we use mercury chloride. And which fixative reacts with lysine residues reversibly? It is formaldehyde. What is the preferred fixative for whole brain autopsy specimens? Perfusion fixation is used. So, what buffer is commonly used in neutral buffered formalin? It is phosphate buffer. And what is the concentration of formaldehyde in 10% formalin? It's 4%. What is the effect of prolonged fixation in formaldehyde? It may cause shrinkage and hardening of tissue. What fixative is commonly used for the glycogen preservation? We use Carnolese fluid. Which fixative contains picric acid and is good for glycogen? Bowen's fluid. Which substance causes protein precipitation and has poor penetration? It's mercury chloride. The ideal temperature for fixation? It's 4 degrees Celsius. What does autolysis refer to? It is the tissue digestion by enzymes or putrefaction. What chemical group does benzoquinone react with proteins? The sulfhydryl groups react. Okay. What type of solution causes the tissue swelling? Hypotonic. And what causes putrefaction? Bacterial action. And what does the glutaraldehyde primarily fix? Proteins. The alcohol which is used in the alcoholic formaldehyde, 95% ethanol. What is used to remove formalin pigment? We use picric alcohol or 1% alcoholic NaOH. Which fixative is commonly used for the enzyme histochemistry? We use cold acetone. And which fixative is used for cytological nuclear fixation? We use Carnot's fluid. And what does the perfusate refer to in a perfusion fixation? The fixative solution passed through the vessels is called perfusate. And what is added to prevent formalin polymerization in cold? We can add methanol in order to prevent formalin polymerization in cold. What fixative is ideal for demonstrating cholesterol? We can use digitonin in order to demonstrate cholesterol. And which acid decreases pH and damages the protein structure? It is done by acetic acid. What is the main protein that reacts with mercury chloride? Histidine. What is the role of sodium thiosulfate after mercury chloride fixation? It removes mercury pigment. What does the term freeze drying involve? The rapid freezing followed by drying. What fixative is used at 65 degrees Celsius to affect the DNA? Formaldehyde. Which alcohol collapses DNA structure? Ethanol and methanol. What type of fixative is Zenger's fluid? It's a microanatomical fixative. What is the role of carbamides? It is a type of fixative used in EM which has cross-linking features. Cross-linking fixative. So it is a cross-linking fixative which is used in EM. Which fixative reacts irreversibly with lysine? Glutaraldehyde. What compound is a part of Haley's fluid? Sodium sulfate. Which fixative preserves fat, lipids, etc. for cytoplasmic studies? Champy's fluid. What's the consequence of using hypertonic fixatives? Tissue shrinkage is the consequence of using hypertonic fixatives. What is the vapor fixation time for formaldehyde at 50 to 80 degrees Celsius? It takes approximately 3 to 5 hours. And what acid is in Gender's fluid? It's picric acid. What is the ideal buffer used in immunochemistry, histochemistry? It's stress buffer. What is the term for non-reversible tissue damage by enzyme? It's called autolysis. And what is the source of pigment and formalin-fixed tissues? Acid formaldehyde hematin. What physical agent can be used in fixation besides heat? You can use microwaves. What is the main use of vapor fixation? It is mainly used in case of cryostat sections. And what is the role of post-chromatization? A modern tissue for staining mitochondria.